this video, we show you how to use Juicebox. To open a map, click the File menu and click Open. You will see a list of all the maps published in our recent cell paper. Double-click the map to open, or click the button below, which also allows you to open map Q greater than 30. The map loads and shows all chromosomes. You will see the heat map and the toolbar with chromosome selection, different data views, normalizations, resolutions for changing the zoom level, color range threshold, and Go To. There is also a mini-map that shows you a bird's eye view of the main map and hover text information. Go to a chromosome by selecting each one from the drop-down menu and clicking the refresh arrows. You can use the resolution slider to change the zoom. Note that the mini-map is updated as well. You can change your position in the heat map by grabbing with your mouse and panning. To change the view, go to the drop-down menu. In Observed over Expected, red shows enriched interactions and blue shows depleted. You can also view the Pearson, or expected. Select from different normalization strategies in the Normalization menu. We most often use Balanced, the Knight-Ruiz normalization. You can change the threshold of the map with the color range slider. In the heat map, pixels with counts below the minimum slider pointer are white, and pixels with counts above the maximum pointer slider are red. Clicking the plus or minus buttons changes the range available to allow you to select a higher maximum. When you change the threshold with the color range slider, you can better distinguish features in the heat map. Go to the Annotations menu to load one-dimensional and two-dimensional features. You can select dataset-specific features such as peaks. These are overlaid on the map. Hovering your mouse over the feature gives information about it. You can freeze the hover text by right-clicking. Another two-dimensional feature available on some datasets is contact domains. These appear as blocks along the diagonal. Once more, you can see information about the features by hovering over them. You can also load Encode tracks from the Annotations menu. Filter the tracks by typing words that appear in the track description in any order. In this case, we select GM12878 CTCF peaks. The track appears both above and to the left of the heat map. Right-click the track to see customization options. Now we go to the Bookmarks menu. You can click Save Location and enter the description for the saved location. The location will be immediately added to the menu. The locations will be preserved after quitting the program, so you can quickly return to the location you are most interested in. You can also clear the locations. Right-clicking on the heat map enables the Straight Edge tool. You can use it to align what you see on the heat map with the one-dimensional tracks. When you right-click, you also see the option to copy positions to the clipboard. You can use these to go to the location in other browsers, such as IGV or UCSC. You can also directly go to locations from the toolbar. Here we see the same location on a new chromosome. We can then save the new location, and then quickly jump back to the old location. From the File menu, you can load a different map as the control map. This is one way to compare different Hi-C maps. In the Views menu, you can switch between Observed and Control in the same location. You can also do so by pressing F1. To see Observed divided by Control, select the option in the drop-down menu. The Open Recent menu is populated with all of the maps you've opened recently. These are also saved between Juicebox sessions. You can quickly access the map you were recently viewing. To jump directly to a chromosome, click on the map. Selecting Show Dataset Metrics gives detailed information about the Hi-C experiment. The Statistics tab has quality control metrics, and various graphs help illuminate the success of the experiment. After zooming in, you might find you want to pan more quickly across the chromosome. You can move the main heat map by larger steps by moving the transparent square on the mini-map. To jump to a different chromosome, you can always use the chromosome selection menus. This overview of the main features of Juicebox should help you get started. We hope you find Juicebox a useful tool for 3D genome exploration. Happy juicing!